Hello, this is Professor Johnson, and in this video we will be talking about some of the basics of cell communication. We'll start by looking at some of the essential types of cell signaling, and the types are all categorized by where their origin is coming from. So in direct contact, the two cells are directly contacting one another, so the molecules can flow freely between the two cells. Paracrine, on the other hand, the cells aren't actually touching each other, but they are nearby. So it starts over here at the cell of the origin. It gets released from the cell and it flows through a small amount of space to another cell that can detect this signal. There's another type known as endocrine that originates from far away. So you might have a cell over here that's on your kidneys, on top of your kidneys in your adrenal glands or within your brain or somewhere. The molecules are getting released from this. It goes into the bloodstream and gets distributed throughout the body and ends up contacting cells. So this is different from paracrine in where the signal is actually coming from. It also has a wider range of effects than paracrine, so you're more likely to see body-wide changes. This is what occurs with most of the hormone signaling. So the hormones that you have, they signal through a type, or this type, the endocrine signaling pathway. The other type that you're probably familiar with is the synaptic signaling. So this is similar to paracrine in that the signal is coming from very close by, but it originates in these special structures known as the presynaptic place, and then the postsynaptic, and then it has the synapse in between them. So it's a very small place. When the molecules are utilized in this fashion, they're known as neurotransmitters. And finally, you can have autocrine signaling. So autocrine signaling would be if this cell right here was to have actually released these molecules itself, and then it responds to them even as an outside. So it's similar to paracrine, except that the cell that actually released the signal was the cell that's also doing the signaling itself. All right, let's talk about some of the parts of signaling. So all signals need to start with a molecule known as a ligand, sometimes pronounced as a ligand, but I prefer the term ligand. So this is the molecule that's sent out or received from the external environment that's going to tell the cell how it needs to respond. So these ligands will interact with receptors. Often these receptors are on the plasma membrane. And then the receptors, when they interact with these ligands, these receptors, because they're proteins, go through a conformational change, and this can change the protein, how other molecules in the cell are interacting with the protein, or it can act enzymatically in a fashion as well. But you're changing how this receptor is viewed by the cell. This in turn can eventually lead to the activation of this effector protein, which will produce second messenger. So the effector protein is distinct in that it's a protein, so it's going to have enzymatic activity, and then the secondary messengers are small molecules such as calcium or IP3 that are going to go on and have their own effects in the cell. We'll look at that in more detail in a future video. Ultimately, this leads to then a second response. Now, it may just seem like it would be easy just to go from one step, the ligand responding to the cell, and then the cell just responding in itself. What's the point of the effectors and the second messengers and the downstream response proteins that were really indicated by this green arrow here? Well, the idea is amplification. So as you go through this, the signal gets bigger and bigger, And so you can start with a very small signal, just a single molecule being detected by a receptor that leads to large-scale effects and a cellular response. The purpose of signaling cascades is to amplify the signal so that you can get a cellular response from just a small change on the external outside of the cell. All right, so types of receptors, we can divide them into two major types, being the membrane receptors and the intracellular receptors. Now, intracellular receptors, you're typically thinking of the steroid receptors, but in reality, intracellular receptors can act in a number of different ways. They can be enzyme linked, or they could be channel linked. So the big difference between intracellular receptors and membrane receptors is that intracellular receptors are going to occur within the cell, while the membrane receptors are going to be attached to the plasma membrane. The plasma membrane receptors can fall into one of these three categories, either the enzyme linked, such as the tyrosine kinase receptors, channel linked, such as the GABA receptors, or the GPCR, which stands for G protein coupled receptors, such as adrenaline. Serotonin also falls into this category as well. The guanylyl cyclase is an example of an intracellular receptor that's enzyme linked, while the IP3 is an example of a channel linked receptor that's actually within the cell itself. And then steroid receptors, you're typically thinking of like testosterone or estrogen when we look at those. So let's look at all these in a little bit more details. So channel receptors work by binding to a ligand. Once they bind to the ligand, you have a conformational change that can open a pore, and then this can allow ions or other small molecules to flow through the cell, thus allowing things to enter the cell that otherwise could not. So that's the basis of channel-linked receptors. Enzyme-linked receptors occur when the protein itself that's acting as a receptor is also acting as an enzyme. So you can have the binding of the ligand here, and then you can have an enzymatic activity. So you saw how that four-pointed star was catalyzed and converted into this three-pointed star by the enzymatic activity of this protein. Intracellular receptors, typically you're thinking of steroid receptors. We'll talk more about those in a future video, but what's really making all of these unique is that the receptors are within the cell instead of on the plasma membrane. So the ligand has to either be produced within the cell, or more frequently it's actually going through the cell and responding. So similar to before, except that you have a response here, or you're having it diffuse through the cell's membrane. G protein receptors are sort of like enzymatic receptors, except instead of having enzymatic activity, it's the alteration of these G proteins that we'll talk more about in a future video. But these G proteins interact with GTP and can actually convert that into a GDP, and that's going to change the activity and how these proteins, or what sort of proteins these G proteins interact with. So you can have the ligand binding to the G proteins. The G protein then, or G protein coupled receptor, it binds to the ligand. The G protein then, in this case, dissociates, and then you can have one of the subunits interact with an enzyme that leads to the production of second messengers, or it can interact with a channel and open up the channel and have the molecules go into the cell. Now signaling, when we look at that, one of the things that can happen is that the same signal can produce different responses. So you have this ligand right here that's responding in this one cell to give this particular cellular response. Maybe it's contraction or something else. It can also act through different receptors, different effector proteins, which would produce different second messengers. All of this could lead to a different cellular response. So you can have one one cell respond in one way and have another cell respond in another, or the same cell can respond differently at different points in time depending on what receptors and what different effector proteins and second messengers are actually present. Now we can look at the converse, where we looked at how the same signaling molecule produced different cellular responses. Now, how can different signaling molecules produce the same signaling or same cellular responses? Well, here we can look at the case where we have two different ligands. So this one over here is responding to this receptor, while this one over here binds to this receptor. However, these two receptors might interact with the same effector protein or different effector proteins that produce the same secondary messengers, which ultimately can lead to a cellular response. Finally, once you have the signal turned on, you don't want the signal generally running the entire time. So eventually you're going to want to shut the signal off. There's a number of ways you can do this. You can remove the ligand, so now you no longer have anything that's activating this signaling pathway, signal transduction pathway. Alternatively, you could remove the receptor, so that you can bring the receptors in and degrade them or store them for later, and then the cell is no longer able to respond to this particular ligand. You can remove or change the effector proteins, which will change or remove the second messengers that are there. Finally, even though you have these second messengers, they can act to these different response protein, which was indicated by this green arrow here, and this in turn can lead to the cell not responding to the message like it normally would. Alright, so that's it for this video. I will see you guys in class. Have a good day.